Hello, my name is Lisette Malposa, and this is my COGs149 Teachback number 2. So this is an outline of today's presentation. I'm going to summarize, highlight key points from the reading selected, propose my independent research, and then give insight and value of my independent research. This is the article that I chose, and it's Evolving the Language and Music Ready Brain. For time's sake, I'm going to only be highlighting certain points of the sections that are going to be relevant to my independent research topic. Um, so this reading starts off by mentioning that there are diverse cognitive domains that ultimately are combined and developed to allow our ancestors to adapt. And to illustrate this statement, the reading goes in detail and talks about no, which is a Japanese traditional performing art. This no contains 3 different components, which I'm not going to try to pronounce just because I don't want to mess it up. So I'll be referring to them by their definition. So this first one, the first component is essentially a dance, and it usually consists of slow and sometimes quick movements. The next component involves the chanting of text, and then the third component is the music that accompanies all the other components. The author then goes on and proposes that there might be a common underlying function or mechanism shared between language, music, and dance. Um, these all can have are individual components that can be modified to best fit the culture and social environment, which is why they stated that even though they can grow and evolve independently, they still have this common origin and beginning. So the following questions that are asked in this next section is regarding the biological, the cultural, and then both of those two evolution things together. So the first one is, how did biological evolution yield a language ready and music ready species? How did cultural evolution yield diversity and language of musical genres? And lastly, to what extent did biological evolution, which yield the language ready brain, also yield the music ready brain? So, the reading mentioned that spoken language evolved directly from the vocalization system of our common ancestors, which are the monkeys. And there are 2 guiding hypotheses for the mirror neuron system hypothesis. So there is the brain mechanisms for perceptual control of action provided evolutionary substrate for language ready brain. And basically, what this is, in other words, is saying that the brain is able not only to detect language, but it also is able to gather information about our environment as well as other people's actions and our own. The second hypothesis is manual dexterity helps with understanding speech. And from what I understood from this hypothesis is that us primans, we want to be skilled and we want to show our skills, and which is why we seek uniqueness. And the unique thing that we have is the language aspect. And this gives rise to, and we extensively use this manual speech gestures while we talk. Um, and then towards the end of the section, the author argues that the language ready brain resulted from the evolution of progression mirror systems and linked the brain regions beyond the mirror, that made the full expression of functionality possible. So my topic of interest, like right off the bat after reading like the first time through, was the mirror neurons. Um, some of the questions that I instantly asked was, what are mirror neurons? What neurological structures are included when talking about mirror neurons? And how can mirror neurons be connected to music? So this is the article I chose, and it's named "What We Know Currently About Mirror Neurons" by Kinner and Lemon. This paper is a mini review, and it captures what has been done regarding mirror neurons and the certain findings and experiments that gathered these findings. So it starts off by defining what mirror neurons are, and essentially mirror neurons are a class of neurons that modulate activity both when an individual executes a motor task and when they observe a similar or same act being done. The goal of this paper is basically to just provide an overview about all of the reviews of the studies that have directly recorded the MN activity of the macaque monkeys. And all of these papers included were 25 total, and they were papers done up from 1992 all the way until this year. This paper was published, which is 2013. Out of those 25 papers, 15 of them found and described the mirror neuron activation to be found in the rostral division of the ventral premotor cortex, also known as the area F5. Um, Out of those 15 that have found that F5 activation area, 11 of them provided an in detail number of mirror neurons that were recording during the experiment. So here there was like 33.6% of F5 activity was described as mirror neurons presented um, once the experimenter was reaching and grasping an object with their hand. So in total, by getting the overview of these articles, they found that activation of mirror neurons established a three interconnected subdivisions of F5. So there is the F5C, which is the connexivity of precentral gyrus adjacent to the inferior limb of the accurate sulcus. There is the F5P, which is connected to both the posterior parietal area, AIP, which is the anterior intraparietal area, and the premotor cortex M1. And there's also the F5A, which is in the depths of the sulcus with prefrontal connections. And then this is right here, so you can see this is a graph that is trying to show a correlation between the publication year along with the percent of the neurons found. Um, just by looking at this, we can see that there has been a great increase from like when the article was published and the percentage of mirror neurons that were activated and found. Um, this just kind of is surprising because it's like now we're in 2020. That's like a 7-year difference, and like just how much more neuron activation has been discovered since 2013. So since I couldn't find any macaque monkey brains, I resorted to showing the human brain just because the macaque monkeys and the human brains are a lot similar in the structure for the most part. So here, these are some brains that I found from Wikipedia, like just Google search. So this is the precentral gyrus right here, and this is representing the F5C right here. And there is the intraparietal, is this red right there, that is representing the F5P. And some of the significant findings that were pretty interesting to me as I was reading it was these sections right here. So it was the real action versus video action, tool versus no tool, relating the human neuroimaging data to from the monkeys to humans. So the first one is real action versus video action, and for this they wanted to see if physically seeing the video produce the same mirror neuron activation as seeing it in real life versus video. Um, so surprisingly, the F5 mirror neuron activation was there for both. There was 26.9% of neuron discharged when seeing the video, which is lower than the actual real action. However, it was not significantly lower in order to say that the real action produced more mirror neuron activation than the video action. There is still a great amount of discharge and mirror neuron activation in both. So there's no one greater, there's no one less. Then we have the tool versus no tool. So two papers discovered that, oh, sorry. So for the tool and no tool, that's basically they wanted to explore if me using a tool to pick something up would results in mirror neuron activation versus me physically picking it up. So, two papers found that there is no discharge of mirror neuron activation, and two papers found that there is mirror neuron activation discharge. For these papers that found yes, the monkeys were shown how to use the tool in a training period before the actual experiment. So this kind of suggests that with enough training and like how to use the tool, mirror neurons are basically able to extend to the tool and not just stop at the body. And then finally, this last section is kind of talking about it's an amazing turning point to my next article that I chose. So this section discussed how fMRI experiences have shown that there is an overlap between the cortical areas in humans during actions observed and the areas in which the mirror neurons in monkeys were reported. So there is an overlap between the mirror neuron activity. So now this is my next article that I wanted to extend. So from seeing the monkeys and all of that, I wanted to extend it to humans. Not only just that, but I wanted to see what about humans, and instead of picking something up, how can it be attributed to music? So the this is the article I chose, mirror neuron activation of musicians and non-musicians in response to motion captured piano performances. And the purpose of this is they just wanted to take a novel approach to studying mirror neuron activations by employing a combination of sophisticated motion captured piano performances accompanied with their musical segments to try to identify the difference in regional mirror neuron activation for musicians versus non musicians. So for each, they had two different musical notes, which is the correct mode and the enjoyment mode. So for correct modes, they were seeing a video of someone that emphasized the technical correctness of performing that. And the enjoyment mode was when the performer can be relaxed and enjoy performing the music. There were two hypotheses provided by the author. The first one was that for musicians, activations in the mirror neuron regions will be greater than non musicians. And for the musicians, their mirror activation would be more evident when they were observing pieces in the enjoyment mode as opposed to the correct mode. So there were 8 participants total. So there was 4 classical piano performer participants and 4 non-musical participants who showed interest in classical music. There was a total of 16 different motion captured recordings, and these recordings were either sections from Coward's song by Edvard Grieg and the Scherzo by John Hummel. And basically, these pieces were played in the 2 different modes that we described previously, so the correct and enjoyment mode. And the motion was captured with an infrared camera system. And this is the reason why they chose this novel approach is by using this infrared camera system, just because they didn't want there to be a confounding variable that was interfering with what the main goal of the study was, which is to focus on the motion and the gestural aspects of the performance, as opposed to focusing on the facial expressions, the hair, what they're wearing, and etc. So they also asked 12 questions throughout after the performances. On a scale from 1 to 7, they had to respond. So the actual fMRI results were that musicians did have, just how their hypothesis, musicians did have significantly greater activations than non-musicians for enjoyment mode presentations. And some of these areas included the left and right supplementary motor areas, right somatosensory association cortex, left middle temporal gyrus. And this is represented by this number one right here. This is the enjoyment mode for the musicians. The next is the correct mode. So musicians had greater activations than non-musicians in the right somatosensory association cortex and the right medial temporal gyrus. And this is represented right here in this fMRI scan. So there was an overlap between activations for both groups, however. So for both groups, during the enjoyment mode, there was a great activation in the right inferior frontal gyrus and the posterior lobe of the cerebellum. And for the correct, there was the activation in the left angular gyrus and anterior and posterior lobes of the cerebellum. And for the ratings, this is the results for the ratings. They only found there to be two significantly two significant questions that from their findings. The rest were not significant. So this was question 7 and question 12. So question 7 is, this performance gave me pleasure. So there was a significantly stronger agreement with the musicians than the non-musicians. And question number 12 is, I am often in situations where I'm listening to music while other sounds, are intruding as noticeably as they are now. And basically, what this question 12 was trying to aim at is the topic about managing your distractions. And here there is a lesser agreement with musicians than non musicians. So, some of the overall major finding points and discussed in the discussion section is that enjoyment mode enjoyment mode produced more extensive motor movements. So, during the enjoyment mode, when the person they were seeing that infrared motion video, they would see that the enjoyment mode produced more extensive motor movements than the control mode because obviously they were told to enjoy the process of them performing as opposed to the correct mode where they had to be technically strategic about how their appearance was. So this also resulted, the enjoyment mode, because it produced more motor movements, it resulted in significantly greater activation of the MN irrespective of music level. So mirror neuron in conjunction with the limbic system is thought to contribute to creation and detection of emotional expectation. So this kind of has to do with the limbic system just is basically in charge of like kind of like with emotions and stuff. So it's pretty clear how the mirror neuron when it is related with the limbic system, it has this emotional expectation. A musician level of expertise and experience produce an enhancement of activation of mirror neurons. And some of the limitations of this study and some of the future studies I'm going to be presenting on is right here in this provided in this section. So one of the limitations is that the number of participants were limited, meaning that they only had 8. So I mentioned before they had 4 in each category. So for future studies, they should include a larger sample size. Try to use other instruments other than a piano, like for example, violins, clarinets, stuff like that. Um, Investigate if mirror neuron activity is relative to expertise. And for this, it's kind of talking about professional versus non-specialist that doesn't get paid. So what I thought about is like two professionals basically that pass like the professional stage, but one that is paid and one that isn't. So now I'm going to provide insight and value of my independent research. So, does your topic draw interest upon other subfields of cognitive science? If so, which ones? And I say yes, neuroscience. This paper is very neuroscience heavy. It contained a variety of brain regions of mirror neuron locations and activations. And I think just the topic itself, mirror neurons, have a very neuroscience and neurobiological background to it. Number 2 question is, based on the methods and results of previous studies, how might you create a new study to investigate your topic? And as soon as like I finished reading these 2 articles, I thought like, how can this be attributed to sports? Just because there is a lot more motor movements involved in sports, and probably the brain activation things would be crazy because everything would be lighting up. But um, a study that I would try to see, like, want to do is try to see the similarities between mirror neuron activation and similar sports that require similar to same mirror um, motor movements. So one example I thought about was the sport lacrosse and hockey. Just they both involve the similar actions of holding an actual um, like tool and basically like doing things with it. So I thought maybe that can be a good um, next, I guess, um, future study to investigate and try to see if there's just significant mirror neuron activation when seeing their own sport, which would probably be my hypothesis that when seeing their sport, more neuron activation would be activated as opposed to seeing the opposite sport, which I am thinking that there might be a similarity just because that tool is used um, similarly in a way, even though the rules are different. But yeah, so these are my references that I cited in my other ones. But yeah, thank you so much for your time. And I